Today I'm going to talk about how it started, Jax's mutation, the turn of events, Jax's status today, and the power of SCN2A community. So we are from Bucks County, Pennsylvania, right out the suburbs of Philadelphia. Jax is going to be 15 years old on August 15th. He lives with myself, my fiance, Jim, and Jim's daughter, Jordan. And if I start to talk too fast, you guys, I'm from Philly. I can translate after if you have questions. So no problem. Last night I was talking and someone's like, I didn't know what you said for 20 minutes. I'm like, we all feel that way. So when Jax was about, I had a pretty good, he was young when it started. The seizure started around 9 months old. At first he looked like he was startled, like almost if you came behind him and were like, boo. So we thought, oh, they're jumps, he's fine, he'll be okay. And then we went and took him over to his pediatrician. We filmed him, everything. We took him to Abington Hospital. They told me they weren't seizures. And we were like, you know what? Something doesn't sound right. So let me go get this checked out at CHOP, because Children's Hospital Philadelphia is pretty close to us. And now that's not working. But anyway, it stopped working. So let me just see if it, next slide, please. Okay, so the beginning of my pregnancy too, I was pretty good, but towards the like 6 months, I started to get something called polydrohydromanose. It was low amniotic fluid, and they were measuring him. He did pretty good, and then he started measuring small, like around the 20th percentile within the 7th and 8th month. They didn't know anything was wrong yet, so I delivered around 38 weeks. And like I said, we took him to the hospital. We took him to his pediatrician, kept telling me it was fine, got to CHOP. They did an EEG at our local urgent care there for CHOP. And immediately they were like, we need to send you overnight downtown. So we knew something was wrong. We got down there and we met with Dr. Eric Marsh. He's the neurogeneticist there. And he ran a couple tests and he saw the writing on the wall. He pretty much was like, this is going to be lifelong. We don't know exactly what. And Jax was meeting, he was still meeting his milestones, but the very, very end of these milestones. So if he was to sit up at, between 7 and 9 months, he was right on the cusp of that 10th month, literally. So after that, his seizures were myoclonic for the longest time. I mean, he had myoclonic jumps up until probably about 6 years old. And then they started transitioning a little bit, and they thought that would never happen. The doctors are like, oh, they'll stay myoclonic. They were pretty sure they would, but they did change. Some generalized, some tonic. It wasn't horrible. We never went into status or anything like that. And he was on Depakote at the time, just Depakote itself. Next slide. We did a lot of testing. So we did not get the SCN2A diagnosis until Jax was about 6 years old. We thought at first he had Angelman syndrome, and we thought he had reverse Angelman syndrome, red herring disease, you name it, we tried to go after it. And then finally, 6 years later, we got the SCN2A diagnosis, and Jax's mutation is a C.3374. He's got a deletion, he's got a truncated mutation, and he has loss of function. Next slide. At the time, there was no one that had this. I mean, literally, the doctors were like, there might be like 3 literature pieces on this. They had no answers. I mean, they really couldn't give us anything about the future. There was really no prognosis, and that he pretty much just had epilepsy is what we were told at the time. Next slide. The other thing is too, they weren't sure about anything for a cure for him, what medications were going to work. And like I said, they treated a lot just like epilepsy then, because like I said, no one, I was calling parents if their kid just had epilepsy and had myoclonic seizures to get anything I could from them, because I was anxious about what the future was going to look like. Would he be okay? Things of those natures. I mean, I'm sure you can understand. Next slide. So in 2015, there was a turn of events for us. Jax was 8 years old, and he started becoming like sick, not eating that great. We couldn't keep the medicine down, and we got real nervous. So we worked with gastro, and they said, let's put a G tube. Let's put a G tube in. And I was on board with it, because at the time we weren't getting anything. He wasn't eating. It was bad. And we were worried he wasn't getting the Depakote. Well, after the G tube got put in, like, I think it was the very, very end of August, 24 hours, we were flown in via helicopter. He was in full septic shock. They had when they flew us in there, they loaded him up with 6 liters of fluid. He was intubated, PICC line, arterial line, and he had full-blown pancreatitis, and they did not know what happened. So we were in CHOP 3 months. I mean, he was intubated for weeks. He then got cellulitis around the G tube, and then they gave him platelets, and the person ate peanuts before they donated the platelets, and he went into full anaphylactic shock because he's allergic to platelets. They told me if he wasn't intubated, he probably wouldn't have made it because they were sticking albuterol and everything into his tube. So thank God at that time, it was a blessing in disguise, but I really thought I was being punked on camera. I'm like, you guys, this is not, you know, if it could go wrong, it went wrong. But then finally around the beginning of November, he started to take a turn. He did better, and we got him out of there, you know, around Thanksgiving. Jax's status today. Jax is now, his seizures had gotten worse. We think it's due to puberty. Around 6 months ago, I told you Jax will be 15 in a couple of weeks. He started with tonic-clonic, the grand mal seizures, extremely bad. He could not breathe, rigid, convulsions, noises, shaking. I had to administer a rescue med about the first time 2 months ago, and we had to take him to the hospital. So we are working on medications now. We had to take him off the Depakote because of the pancreatitis, and we switched him over to Onfi and Keppra, and now we have added in Topamax, but he's a different child now that we added the Topamax in because he's heavily sedated. He usually would be screaming, carrying on, very happy. I mean, he's still there at times, but once we take him off the Topamax, which we tried, the tonic-clonic started right back up. So we have to take it for what it's worth. He sleeps better than he did. We didn't sleep for about 6 years straight. We do sleep more now, so that's a good thing. He is able to walk, but he has a wide gait and he cannot walk long distances. He is nonverbal, but he communicates excellent. He's like a savant with that iPad. So he can play things, you guys. So he'll play like a Jeopardy. My name's Kristen, and if it's just him and I in a room, he'll play like it's just Kristen and I over and over with the voice of like Alex Trebek. My fiance's name is Jim. Jax loves to call him Jeff. So on the iPad, I'll put Jeff. And Jim will be like, stop calling me that. And we'll delete all the stuff that has Jeff on it. And 5 minutes later, you'll hear Jeff. And he also casted SpongeBob Barriass to the 2nd grade classroom at his school to all the TVs at the school from his iPad. They called me and told me he was in trouble. I was like, what did he do? Like, I didn't even know he could get in trouble. But so he's got someone that sits with him now. So he does not cast those dirty videos to these kids. The SCN2A community, I have to tell you, Leah God could not have paired us perfectly up with her at the perfect time. I mean, Jenny, Carla, Michelle, I mean, you talk about excellent. I literally had him in CHOP a month ago. And I'm like, I can't make any decisions. We need to call Leah Myers. I literally had her on speaker with an epileptic specialist, the engine team at CHOP, everybody. Like, I'm like, if Leah says okay, then okay. Like, I'm not making it that she's the say-all, be-all. We love Ben. We spend a lot of time with them. We spend time with families around us, fundraisers, trips, anything. I mean, they're vitally important to our mental health, let alone our physical health. They know the medications. They know the seizure types. They know this fight, you know, and it's a hard one and it's scary, but all you guys that have younger kids, you can thank them. I mean, it's so much better now than it was, you know, 5 years ago. And every year it gets better and better. So I really we depend on them for a lot. But in wrapping this up, the last part of this is I have to tell you guys something with Jax's legs. You see how his legs are very over his head. Well, about 6 or 7 years ago, he fell over our golden retriever. And when he fell, he broke both his humeri. So we roll him into Doylestown Hospital, and his legs were back above his head. And they're like, oh my God, his legs are broken. I'm like, it's his arms. And they're rushing him through the emergency room. They're all checking his legs, and his arms were broken. His legs were fine. But that did heal, and that will never happen again. That was a nightmare. But I want to say it's been an honor. It's been great. And I'm glad to meet everyone. And you guys are all doing an excellent job. So hang in there. Thank you.
It's a pleasure to be here. I can think back to the very inaugural Families SCN2A meeting that we held at Lurie Children's Hospital in Chicago with 40 people in the audience. This is a very impressive turnout, both in person and virtually. So I have the task this morning of trying to explain a very complicated topic, which is what are the functional consequences of an SCN2A genetic variant? Now, SCN2A, as you've probably heard, we call it a sodium channel, which means it's a protein cells use to allow sodium ions to flip through the membrane of the cell. And that gives the cell biological electrical powers. And that's what powers the nervous system. It's what powers your heart to beat. It's what powers your muscles to contract. So these are vital proteins for life. And when even something subtle goes wrong with these proteins, there are potentially devastating consequences, as you know. I've put up some disclosures here because I think it's important to know that as an academic scientist, it's really important to have partners in the drug discovery and drug development industry. And so we have lots of partnerships with groups that you see there. Functionally, a normal sodium channel has properties that lend itself to sophisticated measurements in the laboratory. And the work that was done on SCN2A up until a few years ago was a little bit scattered, and different labs were doing it different ways. Fortunately, Dr. Bender's lab investigated a cohort of SCN2A variants together and came out with the dogma that we are all working with today, which is mutations or genetic variants in SCN2A that activate the channel inappropriately, which we call gain of function. How many have heard this term gain of function? How many have heard the term loss of function? Good. These are prevalent terms, and I'm here to basically try to tease those terms out and help you understand those a little bit better. Now, if the slides do go out, you're going to be forced to watch me do an interpretive dance. So don't make the slides go out to do that. So whatever is happening, please stop it. But the paradigm that we're living with right now, which I think has been extremely valuable for both understanding the biology of SCN2A as well as getting ready to target therapies to different classes of mutations. So on one end of the spectrum in blue are the gain of function mutations, which predominantly seem to be associated with early onset epilepsies and either those that are associated with severe epileptic encephalopathy, as it's called, or maybe even more self-limited forms of epilepsy. On the other end of the spectrum are mutations that cause loss of function, where those predominate in children who predominantly have autism spectrum, intellectual disability, maybe with or without epilepsy. And our question over the last few years has been to ask, are these really the only two functional outcomes of a genetic variant? The biggest challenge has been genetic variants that just change one amino acid in the protein to another one. There's a whole class of variants, and the next speaker probably will introduce you to truncating variants and obvious non-functional proteins. Those are clear-cut loss of function in most cases. It's those single amino acid changes that give us all the heartburn. Those require some kind of experimental evidence to really understand them. So we've to date studied about 130 SCN2A variants functionally in the laboratory. I will skip the details how we do that. A couple of the sets of those variants that we've drawn on are from the Simons Foundation Autism Research Institute databases. The others come from various sources, publications in the literature and other databases, but I wanted to highlight the fact that we have studied 52 unique variants that are represented in the SCN2A clinical trial readiness study. And this is something that Dr. Berg will talk about later, and we're hoping to merge our knowledge on these two topics, from the phenotype to the function, to try to come up with more information. Some variants overlap between those sets. What's shown on this slide is simply a topographical map of the sodium channel protein. In real life, it probably doesn't look exactly like that at all. But these little red and colored spheres represent the location in that protein of the variants that we have studied. Now, on next slide, to understand more about the functional properties of the channel, I wanted to take you through, next slide, take you through some principles of sodium channel behavior, functional behavior. Back up one. Sodium channels exist in different functional states in a cell. Most of the time, they're quiet. They're not allowing sodium ions to pass, and they're in what we call their closed state. That's sort of where they live. If something triggered an action potential in a neuron, that's the electrical impulse that neurons use to signal, upon that trigger, sodium channels pop open, and they allow a rush of sodium ions to enter the cell. That has electrical effects on the cell, and that initiates the burst of electrical activity that we call the action potential. And that's the core electrical impulse that allows nerves to signal throughout the brain. So those sodium channels only stay open for a fraction of a second, and then they slam closed with a mechanism that we call inactivation. And then they stay closed until that neuron or that cell resets itself, and the sodium channels can go back to their resting state. So these are the sort of the dynamic properties of sodium channels. When we measure that activity in the laboratory, it looks like this. We see this combination of squiggly lines. These are recordings of microscopic current in a cell. Tiny amounts of electrical current passing through the membrane of a cell. The downward side of that trace is we equate with the activation of the channel, going from its closed state to its open state. It's conducting sodium ions, it's generating a little electrical pulse. But then those sodium channels say, that's enough, I'm going to shut off now. And then the decay of that current trace is what we call inactivation. Now, we can gain a lot of information from those, just that information. We can actually look at the total amount of current passing in the cell. We refer to that as current density. And then we can also look at the dynamics of the current change and how fast it's inactivating, and is it inactivating completely? And a term some of you may have heard about, persistent current, refers to a small amount of the current that still flows after what we think is the completion of the inactivation cycle. That can be abnormal. Now, measuring current density alone can give us a lot of information. It turns out it's not the most reliable measurement. It's very variable, but it's very clear for some variants that they have a lot more current density than normal. So normal in this case is coded as WT, stands for wild type. That's the normal SCN2A channel. And that's shown here as an example. And below it are these examples of genetic variants that show either larger currents than normal or small or undetectable currents. And so that has given rise to one of the binary classifications of gain of function and loss of function. Now, sometimes it's obvious. There's no measurable sodium current. There's no way to classify that any other way but loss of function. Gain of function's more nuanced. There are some behaviors of sodium channel gating, the opening and closing dynamics, that also give us great information. So here's just a couple of examples of some behaviors that we classify as gain of function. One is that inactivation process is just very slow. And so over time, a cell is being bombarded with sodium flowing in from that non-inactivating sodium channel. Similarly, persistent current, the failure of the channel to inactivate, leaves a small amount of active sodium channels inappropriately conducting sodium into the cell. And this also is what we call a gain of function. Now, these are terms referring to the molecule SCN2A. Sometimes their effect at the molecular level, which we'll label as gain or loss, may have a completely different effect on the cell as a whole. And I'm not going to talk about that. That's a challenge for the future. Now, there are other things we measure that are more complicated. And I may start to lose some of you here. One of the things to know about sodium channels is their behavior is dependent on the voltage in the cell, the voltage across the membrane of the cell. They're called voltage-gated sodium channels for that reason. They respond to changes in the membrane voltage. So we can look at how sensitive the channel is to voltage. Both activation and inactivation processes are voltage-dependent processes. So we can actually look at that. It's a complicated experiment, but here I just illustrate for you what we normally like to see, which is over a range of voltages, we see changes in the amount of current conducted by the cell. As you go more positive, there are fewer sodium channels that are available to be activated. So if we were to see a movement of this relationship to the right, that would tell us that those channels aren't inactivating at those same voltages, and therefore that's a gain of function property. If they went the other direction, that's a loss of function. These are channels that are more likely to inactivate. We look at the same phenomenon with activation. The curves bend the other way. As you go more positive, more channels are available. And again, if that relationship is shifted one way, we can label that as a gain of function. If it's shifted the other way, we can label it as a loss of function. So far, I've already shown you current density, inactivation behavior, persistent current, voltage dependence of activation, voltage dependence of inactivation. Here's another one that's called window current. The list goes on and on and on. Here are all of the things that routinely get measured with one sodium channel variant. So classifying a variant by gain or loss of function becomes an art, because each of these properties could be classified by either gain or loss. And so we've kind of taken the attitude that it's hard to just summarize based on one property. We have to look holistically at the entire channel's effect. Now, I could spend the rest of the day showing you individual sodium currents like this, and no one would get anything out of it. So we've tried to now summarize using simple visual metrics like a heat map, where we color code each property of the channel, each of those 11 properties. Here I'm only showing you some of them, based on a scale from what we would consider gain, blue, or loss, red. And each square for each variant refers to a specific property that we've coded this way. So you can start to stand back and look, okay, let's look at the average variant. They're mixed. They have mixed properties. Now, that's not to say that there's not a dominant effect. It's just that it's more complicated. Now, if we reorder those variants, and these are most of the CTRS variants that I'm showing here, if we order them by going from extreme loss of function, where there's just no measurable sodium channel activity, to the other end of the spectrum, where we have predominantly blue, where they're mostly gain of function properties, you can kind of see there's a spectrum. Just like that first slide, from red to blue, there's loss of function variants, there's predominantly gain of function variants. But interestingly, there's a lot in the middle that don't fall cleanly into either category. Here's some variants that we've studied from the Safari database that are mostly autism associated, not clear that they're all. And this again shows you sort of their profile. There's a lot that are loss of function, but some are mixed. And compare that to a list of variants that we know are associated with early onset epilepsy. And again, there's a mixed property with maybe a little bit more of a predominance of the gain of function there. And so that's the message I'm trying to get across, is that this is a useful paradigm, gain versus loss. We're going to need more information at a kind of a neuron level or a neuron circuit level to really tease out what's the most important driving functional property. But now we can start to do that. So I want to just summarize a couple of points, and then I have something else I want to say in 2 minutes, which is we think this paradigm of early onset epilepsy being associated with gain of function is holding up. It's not 100%, but it's holding up. Similarly, loss of function seems to be mostly associated with the later onset seizures or autism spectrum and intellectual disabilities. But again, not 100%. And so a lot of effort is going to be spent trying to figure out those in the middle. Why are they not falling cleanly one way or the other? So there's going to be a real importance of moving forward with these functional data and comparing it to what we know about the clinical features of those individuals who have that particular variant. So that's a next challenge that we're starting now. Now, I want to thank the people in my lab that did this work, and this work couldn't have been done without generous support from the NIH, funding our Epilepsy Center Without Walls, which is about to enter its last year of funding and not renewable, and also the Simons Foundation for also supporting this work. Now I'm going to pivot to one last thing, briefly, which is it's very clear, and probably many of you have this experience, that not all physicians know about SCN2A. Some specialists don't know about SCN2A. So how do we get the word out? Well, one way is to provide materials that educate people at all levels: medical students, nursing students, genetic counseling students, and people in practice. And so one thing that may help do that is represented in a new series devoted to the genetics of epilepsy. And this is a publication that just got started by Cambridge University Press. This will be a new publication for them. They have a series of publications in other fields that are referred to as Cambridge Elements. These are highly focused mini books on a topic. And those topics range from computational sciences to astrophysics to other things. They've never done one in medicine. Genetics in epilepsy is the first medically related Cambridge Elements project. This got started a couple of years ago with Ann Paduri as the editor-in-chief, and she reached out to some of us who she knew would say yes to be deputy editors. And we also have some other folks importantly involved in this project. The very first issue was an introductory issue. It's available online. Importantly, the target audience here is very broad, and the format is unique. These are predominantly online books that include lots of text to explain things, but also figures and videos and other things that can be displayed nicely online. They will also be printed, but the real focus is to create an online resource. The first gene-related issue is in production now. It's focused on KCNQ2, a gene also associated with childhood epilepsy. But I'm pleased to say that I've now decided to help us move forward to do one on SCN2A, channelopathy. This will be co-edited by myself and Ingo Helbig. Hope he's listening and remembers that I invited him to do that. And there's some key contributors that have been, at least so far, have said yes. They haven't heard what is involved. And there will be some other contributors as well. Some of them are in the audience today. I think the goal of this production is to create a centralized resource that provides soup to nuts everything about SCN2A that we know up to date and present that online. We'll pay for it to be open access so anybody can see it, and it will be a great educational tool, and hopefully this will help to fill that knowledge gap in the field. And so I'll leave it there. Thank you very much.
Set up. Good morning, everyone. Thank you for the introduction. I'm Stacy Cohen. I'm a genetic counselor at the Children's Hospital of Philadelphia in our epilepsy and neurogenetics program, where we take care of lots of kiddos with SCN2A. And today I'll be walking you through the various genetic aspects of SCN2A. So the types of testing that your doctors may have ordered and how to read your genetic testing report and really understand what it means for your child. And there are kind of three major parts to the talk today. First, I'll review some general core concepts in genetics that set a foundation for later. We'll talk about the different types of tests that your doctors may have ordered to try to better understand the cause of your child's neurodevelopmental condition. And then we'll talk about the different types of genetic variants that may be flagged, how they're different from each other, and what they mean. Next slide, please. I have no conflicts of interest. Next slide, please. And we got the outline. Next slide, please. So we'll start with the introduction to genetics. Oh, there we go. So starting at the very beginning, our DNA is the code for our body that provides the instructions telling our bodies how to grow and develop. And this is something that we're born with and that our body uses as we're growing and developing. So our DNA is packaged into chromosomes. And within these chromosomes are what we call different genes. And we have many thousands of different genes. SCN2A is one of them, and they all serve a different purpose in our bodies. So our body uses this DNA code, these instructions, and they first have to translate these instructions into something that's usable by the body, which is called mRNA. It's kind of like the body needing a translation from one language to another to understand. And then it uses this ultimately in order to build a protein. And the building blocks of our body are proteins. And SCN2A is one of these proteins. And these proteins, the code for these proteins is ultimately decided by the instructions that our DNA is giving. So it tells our bodies what building blocks of proteins called amino acids, what goes where and in what order. And then our body is able to use this to make a fully functional protein. So we've talked about how our body uses our DNA, which is packaged into chromosomes, in order to tell our bodies how to grow and develop. And we have many chromosomes in our bodies. We have 23 pairs, or 46 in total. And we know that the SCN2A gene is found on the second chromosome. And we have 2 copies of each of our chromosomes. We get one from our mother, one from our father. So that's why you see on the slide here, kind of each of the 2 lines represents one chromosome in a pair. So typically, when people have an SCN2A related disorder, one copy of their SCN2A gene is healthy and the other copy has some sort of genetic variation. So what is SCN2A? Genes are some combination of letters and numbers. They're not exactly like words, but they're often abbreviations for something else. So SCN2A stands for sodium channel number 2 alpha subunit. And it is one of many proteins that's really important for telling our bodies, or allowing our bodies to have our brains communicate. So we have many cells in our bodies and our brains called neurons, and they need to talk to each other to make whatever we want to do happen. So when we're blinking, walking, talking, these cells need to be able to communicate in a specific way. And that's where SCN2A is really important. And it's one of many genes or proteins that are important in this process. So I've listed a couple other ones on the slide here. So we've talked about how some general concepts in genetics, and then we'll talk about the different types of testing options that are available that your doctor may have ordered and how these tests are different and what they're looking for. So we have our genetics here, we have our chromosomes, and we can look at our genetics in many different ways with different types of tests. So this first test is called a karyotype. That image I showed a couple of slides ago where you saw the chromosomes laid out side by side. That is what a karyotype is. It's basically taking a picture of the genetics and seeing if there's any large missing or extra pieces. The next test is called a chromosomal microarray. And this test is able to look for extra or missing pieces of genetic material, but on a much smaller scale than what a karyotype can see. So it's able to zoom in. And this test is often ordered for children who have developmental delays or autism. It's often the first test that someone is offered when they go to see genetics. And then I've grouped together these last two types of tests because they look at the genetics in a similar way. So our DNA is that code that we talked about. So these tests are zooming in even further now from the ones we just talked about. Rather than looking at the amount of genetic material, we're now looking at the code itself. So what are those letters telling our bodies to do when it's trying to make the SCN2A protein? So a gene panel is focusing on a particular group of genes that have been curated by a laboratory because we know they're associated with a given condition, like epilepsy or autism. And then exome sequencing is a more broad test where they're looking at the many thousands of genes in our bodies at the same time. So a gene panel will often have on the scale of dozens to hundreds of genes, whereas exome sequencing looks at on the scale of thousands. But the idea is the same, that they're looking for smaller changes in the code of the genes themselves that wouldn't have been seen when we're looking at the amount of genetics that someone has. So what are these tests actually looking for? They're looking for what we call SCN2A variants. So a variant is a change in a gene from what we would typically expect, kind of like a typo or a misspelling in a sentence. And in general, most genetic variants are not harmful. We all have lots of genetic variants that just make us different human beings, but as far as we know, aren't associated with any medical problems. But sometimes there are harmful genetic variants, and they disrupt the gene's protein product because of some change in the code of that gene. And so if there's some missing or abnormal SCN2A protein, that affects the brain's ability to use that. So you've gone through genetic testing, your doctor hands you a genetic testing report, and it's going to say, it's going to have a lot of medical terminology on it, but one of the things it's going to do is what we call classify your child's genetic variant, because we know that, as I mentioned, most genetic variants are harmless and they don't cause medical problems. So the laboratory needs to make a call when they see a genetic change. Do we think this is harmful or not? So if something is thought not to be harmful, it's called benign or likely benign. And this is often not even reported on the report itself because it doesn't have any medical implications. So typically when you get a report in your hands, it will say one of these three things: pathogenic, likely pathogenic, or variant of uncertain significance. And we'll go through what each of these things mean. So when a variant is classified as pathogenic by the genetic testing laboratory, it means that this genetic variant is definitely disease causing. And they know that based on multiple different lines of evidence. So there are different types of information that the laboratory can use to make this call. And some examples include if this variant has been seen before in other people with that condition, and is this particular genetic change found in the general population or not. We expect that most pathogenic variants in SCN2A are not found in the general population. Next, we have likely pathogenic variants. So these are very likely to be the cause of the disease, but they're not as certain as it would be if it was classified as pathogenic itself. There's still over a 90% chance that this is truly disease causing. So clinically, we often treat pathogenic and likely pathogenic variants the same as someone's diagnosis. And still there are multiple lines of evidence that the lab uses to classify something as likely pathogenic, just not as much evidence as there are for pathogenic variants. And these classifications may change over time as more evidence becomes available. And then lastly, we have what's called a variant of uncertain significance. And these genetic changes are ones where we know that this genetic variant is present, but we don't know if it's harmful or not. And so again, this classification may change over time, and there are things that we can do to try to better understand the role of this variant. So some of these would include parental testing, if not previously done. We expect that most SCN2A changes that are harmful are not found in either parent. So if that is the case for a variant of uncertain significance, that makes us in general more suspicious that it may be related. And then there's other research testing that is sometimes available just to help us better understand what this variant is doing and whether it's harmful or not. So now we'll talk about the different types of genetic variation that, that you can see on reported by these laboratories. So going back to this kind of instruction analogy where the SCN2A gene is telling our bodies how to make the SCN2A protein. So we have our DNA code. We have our DNA code telling our bodies how to put these amino acid pieces together to make the protein. And there are lots of different places in written instructions where there could be an error. You can have typos throughout a sentence. You can have differences in punctuation, all sorts of things that alter that sentence in some way. So it's important to know how to name a particular change. So that's what we'll cover now. On a genetic testing report, typically you will see what's called the C dot and the P dot. So the C dot refers to the specific change in the DNA code to name that particular change. So this is saying at the 2,558th position in the DNA sequence, at that particular letter, there's typically a G there, but this G is changed to the letter A. So this is how you would read this. And then the P dot is referring to how this particular genetic change is leading to a change in the protein. So in the amino acid sequence, at the 853rd position, there is supposed to be an arginine, but instead there is a glutamine. So this is the way that we name particular changes, so we know how one change is different from another. And so there are different categories of variants. The change that I just showed you is called a missense variant. And that is basically where there is some change in the DNA that leads to an amino acid exchange. So for example, if this is the kind of normal sequence, there's supposed to be this his here, but instead there's something else. There's a pro. So we're replacing this piece of the protein with a different piece. And this is the most common type of genetic variant in SCN2A. We also have nonsense variants. So essentially, this is like putting a stop to the body reading the gene to try to make the protein. So essentially, the protein is supposed to keep going on, but instead, the sequence of the DNA is telling the body to stop reading too early. So it gets cut off and it isn't able to make the full protein. So what this looks like when you're reading your report, there will often be a star here, an asterisk, and that is to indicate that there is a premature stop signal at that position. There's also what we call a frame shift variant, which is similar to a nonsense variant in that it introduces a stop signal too early, but before it gets there, it inserts a couple of amino acids too early. So it has some incorrect amino acids and then a premature stop. And this is indicated by this FS star 11. So this is saying there are 11 incorrect amino acids, and then there's a premature stop. There can also be what we call a splice site variant. So our DNA contains exons and introns, and our body only needs the exons in order to actually make the protein. So it has to cut these introns out. So a splice site variant, you'll often see a plus or a minus, and that's a signal to let you know that it's a splice site variant. And so variants that are called splice site variants are interfering with this process, the body's ability to cut out the parts that it doesn't need. And then lastly, you can have deletions or duplications. These are often larger segments of the DNA that include more genes than just the SCN2A gene, but these can also be found. So why does it matter what type of variant that a child has? Why are we talking about all these different types of variants? And Dr. George just did a much more thorough job of what I can do with the remaining time, but essentially these different types of variants can affect the SCN2A protein in different ways. And knowing this distinction helps us better understand what's going on to actually cause a child's disease. So different types of variants can lead to loss of function and gain of function, as Dr. George was explaining. So I'll kind of skip through this, but the important takeaway is essentially that there are different types of variants that we just covered. And typically what we expect from variants that are nonsense, frame shift, splice site, some missense, and whole gene deletions, we expect an overall loss of function effect. And variants that have a so-called gain of function are missense variants, but they can fall in either category, loss of function or gain of function. So, and as Dr. George mentioned, there are varying different ways that a genetic variant can cause what we call gain of function, but loss of function variants tend to be more similar overall. So in summary, there are various different types of genetic tests that can lead to a diagnosis of SCN2A, including microarray, gene panels, and whole exome sequencing. There are many different types of genetic variants, so it's important to know your C dot, know your P dot, which is to say, know how to name the variant that your child has. And if a variant of uncertain significance is identified, it's important to have a thorough conversation with your neurologist or geneticist to talk about next steps and how likely that variant may be to be causative. And I am a part of the CHOP Epilepsy Neurogenetics Initiative at CHOP. And if you're ever interested in learning more or reaching out to us, our email is there. Thank you very much.
Thank you so much, Stacy. I appreciate that. As we switch over to our next slides, I want to say probably one of the biggest goals of the group of speakers this morning is to eliminate that term variant of uncertain significance, make it a thing of the past. So everybody that comes in that identifies something completely novel, completely new, actually has information from day one. One way in which you could do that, and one way in which you can work short term to come up with predictions with that is through computational techniques that Dr. Dennis Lau of Cleveland Clinic and also of the Broad Institute has pioneered for SCN2A. And so we're looking forward to having him speak here as your slides eventually become available.
Yeah, thank you very much for giving me the opportunity to speak. Let's see. So I'm talking about a lot of software and websites, and if my slides don't work, I have a really hard time, but let's keep the fingers crossed. So one thing what I really want to start with is the following. So at the moment when you came the first time to the hospital, you receive a genetic test result, and you were confronted with a medical doctor, you might have noted right from the beginning, perfect, that there is on your medical counterpart side sometimes a little bit uncertainty. So when, for example, I would break my arm or my leg, a medical doctor or a geneticist or a genetic counselor, they really know what to do, because this is something which is, has been tried and errored for 200 years, maybe even longer, probably a couple of thousand years even. And we really know what to do in such a scenario. However, when we think about SCN2A disorders, the situation is very different. And many of you have probably realized that sometimes if you ask a question, for example, is my son or my daughter, does they have like a gain of function variant, or what does that mean, this word SCN2A? You might have noticed that the answer is sometimes not so satisfying. And this is just by the nature of that we have now new technology in genetics since 10 years, which allowed us to basically diagnose diseases for the first time. But the problem at the same time is that out of our, let's say, 4,800 neurologists, I just looked the number up in the US, no one received training how to handle SCN2A, zero. At Cleveland Clinic, I'm leading since 4 years the education for residents and fellows at the Neurological Institute for Genetics. And they didn't have good training in medical school, the curriculums are old and they don't incorporate these kind of precision medicine and care paths. So, and once someone has received education in neurology, they have to do all these fellowships, which require them to work 15 hours a day, read all the EEGs and so on and so forth. It's really hard for them to learn on top of this now about SCN2A, because SCN2A at the end is not the only gene which can cause really devastating neurodevelopmental disorders and epilepsy and autism. There are actually a lot of other genes which can do this, and every gene has its own knowledge that you have, which you have to apply to do the best care possible. So how do we, get to the physicians and even the genetic counselors, to help in the most precise way possible for every gene? And that's a big challenge. And this is just the situation we are at the moment, where we have to incorporate all this knowledge which we just have gained or currently gaining. Every week there is a new study which might have clinical implication. And everyone is here with a lot of hope, where we think about, hey, we have even companies here who have or potentially have drugs in development which might help your children. And so the whole point is where for other disease areas, there are game plans in place for hundreds of years. For diseases like SCN2A, we are just able to diagnose them. You know, in all humanity, there were SCN2A children even like 100 years, even 10,000 years ago. But only since like 10 years, we have the technology to identify that is the reason. So no surprise that at the moment there is a challenge to incorporate this knowledge into better care. So, and on the flip side, there have been now, since we know SCN2A, there have been now hundreds of studies which added some knowledge, but only in the last 10 years. So we have now a volume of information which potentially can lead for every child a better treatment or better decisions, but how to incorporate this into decision making. And before I talk about potential algorithms which can help, there's you know, always the goal to have, for the person who is caring about your child, either the geneticist or the genetic counselor in terms of interpretation of the variant, but also the neurologist or therapist, to have the best information which was just found. And at the moment, this is in research. There are no agreements at the moment. The book, what Elle just described, is coming, but at the moment, it's a lot of digesting information from research, and there are every month 20, 30 studies on SCN2A, and then make an evaluation and then maybe applying it to your child. And this is something which is really hard, and because not everyone is really skilled and has the time to read the literature and then make the decision what is useful. So long story short, and I don't go much into detail, the website is there, and if you ask someone from the foundation, everyone has it. We tried to aggregate the best knowledge, what we have from the literature, curated by experts from the foundation and friends, to have all information in one place, and its updates on a regular basis. So this is the so-called SCN Portal. And in this website, we not only look at SCN2A, but also at other SCN genes, because we scientists believe there is some meaningfulness in looking also at related disorders. So, and with this, we have information there from more than 2,400 patients with sodium channel disorders, and including more than 400 children or individuals who have SCN2A. So what I personally believe in is, if we have information from, like say 400 children, some statistics to it that can be valuable for a care provider who has maybe never seen a single patient with SCN2A before and starts to Google then about the disease. So that's the whole point I want to make. So there's a lot of literature, there's a lot of information here where this information is provided in the website, and one can be used to look at the data. Okay, so I already have half of the time, so and I didn't even started with my second slide. Okay, so let's go for that. So I encourage everyone from the industry and academic side to make this website better. So to make that very short, and we have here on this website specific tracks. This is some, for example, from the clinical providers, that they can learn about the disease, also for parents. And you know, some of the parents might go really into nitty and gritty and might even look at some more researchy things. And I am always saying the work we especially what our team is doing, that can be taught in over 2 or 3 months to some degree to have a deeper insight. And we are always happy to you know, have interns. If you as a parent or have a child, I don't know, a friend who really wants to learn about the disease, we can teach whatever we know. That's not so complicated at the end. The biggest barrier is just having enough courage to just do it, because at the end we don't know much about the disease that that one cannot learn the basics about it. Anyways, so we have these interfaces where you can learn about the disorders. You have some background information, and we listed the publications which we think are most worth reading if you, as a, for example, physician or a parent, want to go into the science. We have statistics, how different patients look like in terms of the disease subtypes. Then we have info videos, for example, also what are SCN2A disorders. If you want to show this, for example, a family member or a physician, just to get some basic ideas, and we try to keep it simple. And if you, for example, are working graphic design or really are interested in communication, we could use a lot of help in developing videos, why you should enroll in a clinical trial, how good such meetings like this are. So there's for everyone the opportunity to contribute to this resource, so that if someone wants to learn about SCN2A disorders, they don't need to try to find information from 1,000 places, but you have that here organized in a place, which also has then been supported and vetted by other people. So, and about the video and so on, you can talk to Arthur. He's sitting at the very last table here, and he has a poster. So this is, of course, supported by many organizations, including ours here. So the next thing is that we have an interface for genetic counselors, is that, which has all the information from, 400 individuals with SCN2A there, and a lot of information which typically would take a genetic counselor up to an hour to get to all these type of data points. And we have them nicely organized here, available in 1 second. So if you feel that your genetic test report might not be that thorough, or you just have another encounter with a genetic counselor, just show on the webpage. Maybe that's useful for them, maybe not, but I potentially would think it is. So I don't want to go much in more detail, and we can actually use this information to also develop some algorithms which are really useful for genetic counselors to maybe make a decision why some mutations are not these variants of certain significance, that they are actually more likely to cause disease or not. And here's a poster from Tobias and Alina, which you can talk about this more, for example, if you're interested in what makes a variant pathogenic using current guidelines and how we can incorporate algorithms. So lastly, we have also like an interface. This is more for like neuroscientists or structural biologists. So if you really want to learn what a position of a mutation can be correlated with certain diseases, but I don't want to go in this much detail, but you can play actually and learn about mutations and what do they do in this tube, which is called SCN2A or NAF 1.2 at the end. So I'm skipping here ahead, and also we have a team member, Nisha is here, who does molecular dynamics, so actually simulating what the mutation could do, and she can talk about that as well. So I go ahead here now and just describe a little bit what our next step is, and this is just having more data. So every information which comes through research, so that's why I really encourage you to participate in a lot of research studies. We hope to get from the individual scientist, again, from academic, but also from industry, and then add it to this resource, so that for every mutation, we have so much information that because from animal models to specific assessments like the eye tracking, so that we can have a good understanding if we find another patient with that same mutation, what is likely to happen and why. So, and in the new version, we built actually the portal like a platform where clinicians can enter data, and we and we even have a feature where you could in theory upload an MRI or EEG. So moving really into with user accounts and more analytical platform where we also maybe have user boards. So we're moving in that direction. It's an ongoing project, and Jinkui, who is here at the last table, has also posted on this. So very lastly, actually good on time. So So we want to move not only towards data aggregation, where someone can spend time and then does their algorithm in the head to use this information, but we actually want to move to decision support tools. So at the end, the caregiver, your neurologist or geneticist or pediatric neurologist, they will be the people who take the responsibility. That will not go away. And this is also correct, because in every situation, there might be things which an algorithm cannot pick up and needs to be considered when a clinical decision is being made. However, since everyone knows that SCN2A is individually rare, we live in a world where your caregiver, your, let's say, neurologist, it's very unlikely that your caregiver has seen them more than 10 patients. And it's very likely even that among the 4,700 neurologists which we have in the US, that more than 4,500 have never seen an SCN2A patient. So, right, it's really important that we have resources which give a second opinion, and ideally they are based on data. So there was a recent study where we also have been involved, where someone could use a lot of data to predict, for example, if the effect is gain or loss of function. So as Dr. George earlier mentioned, we can test many variants, but I think for the SCN2A genes, at the end, there are, I don't know, 10, 20,000 variants possible. So we need, so that's really challenging to test them all, but actually technically feasible. If you are a billionaire and talk to Dr. George, he can do it, even in a year. So, but at the moment we're living in a world where this is not happening. So, and that way, but we can take the limited data we have and can try to generalize from that. But and we are moving in the head and we are doing this. But the thing, what I want to stress is, when we want to predict the gain and loss of function, then we are at the moment round about out of 100 tries, we are 73 times correct, but we are also 27 times wrong. And, you know, and this is not good enough if you want to go to clinical trials or so. That would be way too much of a gamble. So, but with more data we have, which we can use for these models, we might get to better algorithms. And this is really why I encourage you again that we generate more data to do such research. And furthermore, we know also that the binary, so this classification gain or loss of function, if you look at the room and the children, that's not so, that's an oversimplification. We need to develop better algorithms which can stratify in more detail. So again, so we need to generate more data. The existing data needs to be shared, and there's a lot of data which we don't have, which is existing. There's so many natural history studies going on, and none of them is in our portal, just as an example. And we can build better resources and algorithms with more data. So the last thing I want to show, where I feel pretty proud and also I feel very strong about, conflict of interest, of course, because we have done this, in particular Eduardo from our team. So this is a study where we work together with many people across the globe who were seeing people with SCN1A disorders, a very similar disease like SCN2A. And all these collaborators who have seen the patients shared with us data, so that we had more than 1,000 patients with SCN1A disorders together. Just as a reference, we are with SCN2A, we are at 400 at the moment. So, and for these 1,000 individuals, we looked at their clinical data and we generated a new genetic score. So, and what we noticed is that individuals with SCN1A disorders are very different when it comes to when the seizures start. So this is one version of the epilepsies, and at the end it's a little bit more complicated. But in this study, we compared a mild version of the epilepsy versus a very bad catastrophic one with number of many seizures and also associated with unexpected death. It's so whereas the mild disease is people can actually have a relatively normal life. And the biggest question now for treatment is early on, is it the mild or the bad disease? Because for the mild one, you don't want to do anything, but for the bad one, you want to intervene as hard as you can. But as you know, all these drugs which we give and potentially also the gene therapies, they have really strong effects. And if you don't want to give it to the mild disease. So what we learned is that the mild disease actually a super wide first time of seizures. Whereas the bad disease, basically almost everyone has seizures around month 7, a little bit, let's say 2 or 3 months before and after. And then whereas the mild disease, it's from month 4 to month 2. It's a very different picture. And and it became only really clear as cut when we looked at the 1,000 patients. Before there was some evidence like this, but it was not that crystal clear. And what we also noticed is that the genetic variants look a little bit different, which we can also capture a little bit. And the interesting question was then, can we combine this information in a very simple statistical model and then make a prediction out of this? And this is what Eduardo did. And what we learned is that if we then test this model, and we were around about 9 out of 10 times correct with predicting if a patient would develop the mild or the severe disease. And when at that point we thought, okay, that would be nice, but do we feel strong about it, or not? And one way how scientists feel a little bit more strong about their predictions is if they give it to colleagues and they don't look at the results and they just test something blindedly. And we did this twice. We replicated these kind of predictions, one time in Australia and one time in Belgium in a blinded way. And both times we were 9.5 times out of 10 times correct. So this information is now entered into a website where someone can, for example, put in the seizure onset and the mutation, and then you get a probability if you will develop the mild or severe disease for this SCN2A disease. And that has been double-blindedly validated. So this gives me a lot of confidence that this is a good second opinion. So compared to an individual, compared to a provider who may have seen 1 or 2 patients, or maybe even non-patients with that disease, if that person has this in hand to, confirm their intuition, I feel more strong if that such an individual would make a decision without such a resource. And the point I want to make is this resource is built on data which was existing. That no data has been specifically generated for this algorithm. So there's only the seizure onset and the genetic score, which bioinformaticians like us can generate. It's probably a little bit more complicated for SCN2A, but this means it takes maybe a year longer or 2. So if we have enough data. So, and we can plug in also functional data and so on and so forth. So long story short, I really want to acknowledge everyone who shares data and encourage everyone else to support this project. And literally everyone can contribute. Even if you just send me an email and say, I hate the website because I cannot understand this. This is really helpful to improve it. I'm serious, right? So, and if you have like a super graphics, someone is working in marketing, communication, or you just have a nerdy child who's really want to do stuff, we have jobs to improve this. So with that, I close and thank everyone.
All right, have a seat.
Okay.
All right, wonderful. We're going to have a little session right now. If Stacy, where'd she go? There she is. She's going to come up. If anybody has questions that they want to write down, we have some, they're going to be coming around. Just submit them. We're going to have runners and heels. Oh man. Okay. Don't run too hard. And we're going to ask some questions. I'm going to start it off and I want to ask you a question. We talked a lot about missense variants this morning and how it can be extremely hard to understand what an individual missense variant does and how it changes. But some of the families in this room have loss of function variants, or I would say presumed loss of function variants that are associated with protein truncation. How do we think about those when we're thinking about genetic diagnosis and child's care? And we do have a mic that we can share.
So when we think about these presumed loss of function variants, there are lots of variants out there to study. And the ones that are frame shift or nonsense variants, we know kind of cut the protein off too early. And because of that genetic mechanism that we know about, that's kind of why we say presumed loss of function from that variant, even if it hasn't been studied in a lab necessarily. And so when we know this, there are certain things that we can say in general about what we expect. So typically children with these variants present with autism or developmental delays first, and they may develop epilepsy a bit later into their childhood, but many don't. And so that's why when we say presumed loss of function, we're able to kind of presume that from what we know about the genetic variant and how it affects the protein, and then can give some general guidance there regarding expectations based on what we know about other people and families with those variants.
Okay, great. Thank you very much. Actually, this question has come up several times here. And so I would actually like everybody in the group to comment on this. So For example, if someone has a protein truncating mutation, are there any abnormalities in the function of the sodium channels, or do you just have a decreased number of normal channels? And I'll leave that one for Al. If you have haploinsufficiency, 50% of the normal 1.2 channel, are there any abnormalities in the function of the channel, or do you just have a decreased number?
Okay, so an example, what's the new term here is haploinsufficiency, which is half the number of normal SCN2A gene products. There are many ways to get there. In Stacy's talk, she had a list of types of mutations that lead to complete loss of one copy of SCN2A, loss of function of one copy. For example, the whole gene could be gone and deleted, or part of it could be gone, or it's misread by the cell. So at the end of the day, there's no functional protein. So there's an example where haploinsufficiency really means there's only one functioning copy of the gene. The other one is just not there at all. There are scenarios where we would say there's functional haploinsufficiency where the protein's made, but it's not working in some way. Now, we judge that based on experimental systems that are not a human brain. So it's always possible that there may be some effects in a cell that we can't anticipate or we can't detect using the limited experimental system that is normally used. Does that seem to answer the question, you think? Does that sound okay? Okay, I have a. Okay.
I'm the guide. I can't pontificate right now.
Yeah, maybe just to add to this. So one of the biggest clinical differences, what we observe between individuals with these so-called gain of function or haploinsufficient disorders, is that the seizures start on a group level and at very different time. And what we observe if you compare those mutations which have been seen in people with seizures which start directly after birth, and if we compare those mutations where people have this truncating variant and those where they have these missense variants which also lead to loss of function. The ones where the missense variant is leading to loss of function and they have a truncating variant, they have the same seizure onset at the same round about the same time, whereas the one which have gain of function mutations, the seizures start very early. So that makes us to believe that these truncating variants behave similarly to a channel which is there but is not functioning anymore. So these missense variants, which we call loss of function.
Yeah, to add to that a little bit, there have been studies in mouse models, rat models, and prairie vole models, and I'll explain why you would want to study a prairie vole, but there are SCN2A loss of function prairie voles that exist in this world. And under all of those conditions, when we have this case of haploinsufficiency where you've lost half of the genome, one of two copies, the neurons in those animals behave as if there are half as many NAV1.2 SCN2A associated channels. And the other half is still there and functional. It's just that they've lost that one half. Now, related to your question, or related to your answer right there, Dennis, there's a related question about loss of function genotypes. And if you have a particular variant, let's say a missense variant that is associated with loss of function, is there enough information right now to then predict the age of onset of seizures or the severity of those seizures, or whether that child will ever develop seizures that would typically be onsetting later in life, within probably past the first year? And I think the answer is no.
Yeah, so so what we have to distinguish is if we look at things more that we have, if we can make an educated, like if we can make a guess, we can make some guesses. But if this is something where I would give my left hand and I'm using my right hand for important things, no. So so on the, there are many things which could influence an individual disease direction or trajectory, and we don't have enough information to have strong expectations. But I would always recommend to discuss this with your individual caregiver and not generalize from these group level observations we make. If the question is, are we there maybe in a couple of years from now? And you know, I'm a very optimistic person. I would say yes. So for will we be always for every child? No. But I think for many variants or like mutations, and also with the additional information, what drugs have been received so far, how did the disease develop before, were there complications or not, we can probably have good estimates in which direction the disease is likely to develop.
Great, thank you, thank you. There is one question that I'm going to read off, but I'm going to also say it's very appropriate for our later afternoon session, and that is once we understand the biophysics of these channels, how does that affect the functional impact of other ion channels within a neuron or the overall excitability of neurons? And Yan is going to talk about that in detail this afternoon. Jennifer Carney is going to talk about that, and I'll talk about that a little bit. So we're going to table it till then because we have pressing questions here. And that, okay, here's a great one. Let's say you have a variant of unknown significance. It's a missense variant, and seizures started on day 1. Does that make it likely that it is gain of function?
Yeah, I think there would be general agreement that that's likely. That seems to fit with what we know about the clinical genotype phenotype correlations so far established. And again, I think the probability of that is being very high. Would I give my left arm to do that? Yeah, maybe a finger. So
Okay, John Wick.
Just to add to this,
and related, I want to add something to that. And related to that, does that help us understand which medication should be considered first in line?
Yeah, and just one thing to add, because if it's a variant of uncertain significance, one should not act on this. So if that the question is only, does this information maybe convince the physician or genetic counselor to say it's more likely to be SCN2A, then this information might also then trigger that it's likely gain of function. But if for some reason the variant is classified as VUS, I would really discuss with your counselor how certain that is, because you could also have like neonatal seizures because of KCNQ2 or other reasons. And so just to add to this very briefly.
again, the question was about whether that influences treatment decisions. I mean, again, I think knowledge of a sodium channel variant early in life, if a physician wants to go down that pathway, there are lots of sodium channel blockers. The question ultimately is which one to start with. And I don't think there's any good guidance about that. You know, we've been trying to do in vitro pharmacology on genetic variants. And, you know, there are some variants that respond differently to different drugs. Whether that's going to be able to be predictive of how a patient responds, we don't know yet. And I think in the long run, we'd love to have a set of a data set of drug responses of individuals with known variants that we can then test in the laboratory with those drugs and other drugs to see if this could be predictive. Ultimately, that would be a great advance if there was a laboratory test that could say, here's the genetic variant that we just detected, which drugs would work best. That's a dream. We really need to verify that the laboratory test predicts a clinical drug response. We don't really know that for very many variants at this point.
Yeah. And I'm going to be paraphrasing a couple questions here. We've just talked about medications associated with gain of function seizures. There is a question about seizures associated with presumed loss of function variants or loss of function leaning variants. When do those seizures typically start? And are there guidelines associated with medications there? Should you choose sodium channel blockers or avoid sodium channel blockers, and what are the alternatives?
Yeah, I think if you look at around about 100 or I don't know the exact number which we have, for example, in the portal, you could actually test it yourself. You can go to the website and select protein truncating variant and look at the seizure onset. It's on the basic information page. And I think the average seizure onset across individuals is around about 1.5 years in that realm. But you see some individuals have still seizures around 4 months, but it's only few if you have a protein truncating variant. And really the majority of individuals is around 1.5 years to 2 years. There is some evidence in the past from another ion channel disorder like SCN1A, where the lesson has been learned that you should not give sodium channel blockers if there is a truncating variant. This has not been validated as far as I can tell for SCN2A, but this is something definitely to discuss with your care provider. I would just bring it up, and just as a point, there's no stupid question. So because we are living in a world, as I mentioned before, where research is very close to knowledge. And if you read about that sodium channel blockers could be, or hear today that might be a concern, I would discuss it with your care provider. Yeah.
Great. Okay, so now we get into a question of prevalence. And when you think about the entire population, both people that have been identified and people that have not been identified, what do you think the relative percentage of patients would fall on the gain of function side of the spectrum or loss of function side of the spectrum when you consider all missense variants and protein truncating variants? What are the odds if you just took a sodium channel?
I think we're about to calculate that. So I'm not really, so based on the numbers, I guess what we observe at the moment, but there's biases, of who you see. I think it's 2 to 1 in a ratio from gain of function to loss of function variant carriers. But there's a big bias because I think the gain of function patients, they are more likely to be identified.
Yeah, that's in terms of ascertainment.
But just in the mutation rate, you have way more missense variants than truncating variants. Just and then there's this. But still, anyway, so we calculated this a couple of years back, but my guess would be 2 to 1. That's my guess.
Oh, you have a comment?
Yeah, I think what Dennis just said, which is missense variants, way outnumber other kinds of variants. And unfortunately, that's a class of variants for which we have less certainty about what the functional consequences are in general. And so I think it's a hard question to answer.
And we also have this kind of third category that Dr. George was mentioning, these kind of mixed presentations too, and that kind of throws a wrench in these kind of general numbers. And we have the distinction between kind of overall effects and different ways that a variant can lead to both gain and loss of function effects. And then we don't know exactly yet how that plays out in the brain itself. So it makes it tricky because when we're using this dichotomy of gain of loss and function, we're missing some of the nuance there.
Okay, great. I have 2 more things I want to cover during this time. And the first is probably the most important question that we have here. Dr. Lal, what is the best way as a family to contribute to the Portal data set?
Yeah, probably what I guess just communicating what was helpful might be good, but it's definitely more productive to communicate what was not helpful, what you wish should be there and what was not there. And not everything will end up in the portal. Some could inform also how the family and foundation sites are improved, but definitely just giving feedback. And if there's opportunity to even make suggestions or provide, volunteer with help to improve certain things, that would be even better. So, but you could either you have a skill or you can just say, hey, I have time, and there are certain things which could help. But otherwise, spreading the word, and I would definitely bring it up and schedule time with your care provider or your genetic counselor look at the website together. They should have some ideas and maybe give us feedback that we can improve it. If you speak different languages, we have now included or we are about to include an Google API, which translates automatically the website in all possible human languages. But that's has struggled with genetic terminology, right? So if you speak another language, you can help us a lot just translating the web page. But in general, just play with it and not hold back, give us tough feedback.
Okay, wonderful. Thank you so much. Let's give a big round of applause for our panelists here.